Hello, my name is Paul Gloth, and this is my presentation discussing the virtual core flow cytometry paper by Solwell et al. Before discussing the paper, we have to look at what flow cytometry itself actually is. And typical flow cytometry is trying to determine physical and chemical characteristics of a substance through refraction of fluorescent light. And it is done by putting the desired substance into a sheath fluid that is funneled through a nozzle and will typically come out at one cell or one part at a time to be funneled through the laser, where the laser will cause fluorescence to reflect off the cell that is measured by uh, different tools and analyzed to see what is what is actually fluorescing on the surface of these cells or other substance. The problem that this paper discusses is that most flow cytometers work very well in a laboratory setting where you have a large quantity of sheath fluid to run through the system, because it typically takes a couple of liters at a time in order to do accurate and correct measurements. And this paper wants to approach an idea that could be used out in the field or more readily by locations that had less available resources. And in order to do this, they created a flow cytometer that had what they called a virtual core, where instead of using a large quantity of sheath fluid and flowing one cell at a time, they used a more diluted version of the sheath fluid that allowed them to pour the substance into a tube and measure the refraction from cells that were traveling through the center of the tube, rather than having a completely focused refraction or flow of the cells. In the first image on this slide, you can see the setup of their virtual flow cytometer, where they have light being objectified through a lens and focused to a single pinpoint, where the areas around the pinpoint are mirrors that will relay what is actually being aimed, or what they're actually aiming, to a camera so they can properly focus and aim the uh, the laser that's being shot through the substance. And they also have two detectors on the left and right with accompanying mirrors that will detect the refraction of light that doesn't hit directly on the center of their virtual core that they're looking for. And then they have various sensors on the other side of the refraction, or on the other side of the pinhole, that they use to perform the same measurements that a typical flow cytometer does. The second picture depicts their methodology in showing that a proper refraction will allow them to measure the data that they're looking for through the right way. And they also show that they can differentiate between the cells that are passing along the same line of the laser but that are not within the virtual core. And part D of the second picture just shows those small refractory mirrors on the side that go to the two detectors on the left and the right, D1 and D2. In these plots, we can see the results of their new virtual flow cytometer, where in the first plot on the top left, the part A shows the results of looking at T cells labeled for CD4 and CD8 flowed through a typical flow cytometer, and B shows the sample being sent through this new virtual flow cytometer, and then C shows the same sample as B, but with a filter applied, where you can see that it breaks it down into the three parts that you see from a normal flow cytometer using regular sheath fluid. And in the paper, they discuss that this is actually only a 2% difference in observed ratios of CD4 to CD8 from what you get using a regular flow cytometer. The second image shows flow cytometry of raw sewage looking for bioluminescent or photoplankton, and you can see again that both markers that you see in the flow cytometry, typical flow cytometry, are seen in the virtual flow cytometry, and the 1 micrometer and 6 micrometer beads that were put through for calibration can also be seen using the virtual core flow cytometry. This paper discusses in its conclusions that this new virtual core flow cytometry can show as little as 2% difference to typical flow cytometry. Therefore, it can be comparable to your typical flow cytometry in determining the physical and chemical characteristics of your samples. It is significantly more efficient with sheath fluid. While it does require a small bit, it is a lot less than a typical flow cytometer. And it is significantly less, or not significantly, but it is less complex and would be more suited for field work there where you don't have all of the equipment readily set up as you would in a laboratory setting. These are my references for this presentation, and thank you for watching.